Assalamu alaikum. We're here to conduct and demonstrate 4 important practical experiments under the course of seed science. These experiments are essential for evaluating seed quality and understanding the scientific principles behind the seed testing and certification. The 4 practicals that we will demonstrate today is seed moisture content test, seed purity test, seed germination test, and seed electrical conductivity test. I am Sajadul Emon, ID 242555089. Now I am going to demonstrate the test of seed moisture content by 2 methods. One is destructive and other is non-destructive. The destructive one is the seed grain moisture meter by crushing method, and the other one is seed grain moisture meter by gravimetric method.
At first, we have to take some seed samples into the petri dish. Then we have to place some seed samples into the plastic tray. The next step is to insert the plastic tray into the machine and begin to crush. We crush as tight as possible and wait few seconds. Then the seed moisture content is ready to watch. It displays that the seed moisture content is 13.7%. The second one is to determine seed moisture content by using gravity. At first, we have to switch on the GMM mini machine. The next step is we will place the plastic tray and begin to pour the samples. We wait and fill the cup with the seed sample and level it properly. The next step is to remove the plastic tray as quickly as possible and wait few seconds. Now we can see that the seed moisture content is ready to watch, which is 12.9%.
Assalamu alaikum everyone, myself Sakibul Ratul, ID 242555074. Today I will be conducting an experiment on seed electrical conductivity, also known as EC test. EC test is one of the most basic seed tests to determine seed quality and vigor. Seed electrical conductivity measures the amount of electrolytes leaked from the seed when they are soaked in deionized water. When seeds are weak, aged, or have weak cell wall, they release more ions such as potassium, calcium, manganese. Now I will be conducting the experiment.
Select healthy and uniform cauliflower seed. Place the selected seed into a mortar and pestle. Gently crush the seed slightly to expose inner tissue. Do not make powder. Wash the EC meter probe through with distilled water and wipe gently with tissue paper. Add a measured amount of distilled water into the flask. Transfer the crushed seeds into a clean conical flask. Insert the EC meter probe into the flask solution. Wait for the reading to stabilize. Record the electrical conductivity value.
Assalamu alaikum. I am Ratul Bhuyan, ID 242555090. In my portion, I will cover seed germination test, which is one of the fundamental tests used to evaluate the seed quality. The main objective of this experiment is to determine the germination percentage of given seed sample. Now I am going to demonstrate the procedure of seed germination test. At first, healthy and suitable seeds were selected for the experiment. Then, the selected seeds were placed in a petri dish. After that, 50 seeds were counted separately and transferred into another petri dish. In a different petri dish, blotting paper was placed and arranged properly. Then, 7 to 9 ml of distilled water was added to the petri dish containing the blotting paper to moist it. The seeds were then spread uniformly on the moist blotting paper, ensuring that the seeds did not touch each other. After placing the seeds, the petri dish was covered with its lid. Finally, after 24 hours of observations, it was found that the seeds had germinated.
Assalamu alaikum. This is Homayun Kabir ID 242555086. Today I am presenting this video as a part of our seed science course. This topic of my experiment is seed purity test. Seed purity test is conducted to determine the percentage of pure seed present in a given seed sample. In this test, the seed sample is separated into 3 components: pure seed, other crop seed, and inert matter. Now I am going to demonstrate the procedure of seed purity test. First, pure seed, inert matter, and other crop seed were taken together in a glass container and weighed using an analytical balance. Then the component, pure seed, inert matter and other crop seed were separated carefully on a tray. After that, the weight of an empty glass container was measured using the analytical balance. Next, the separated components were weighed individually. First, the pure seed were placed in the empty glass and weighed. Then the inert matter was weighed in the same way. Finally, the other crop seed were weighed separately. The final result of my experiment, here is the pure seed 82%
and other crop seed 11% and inert matter 7%.